Okay, so this is our early vertebrate digestion and respiration lab coupled to the integument. And really the rationale is to try and get a picture of some of our early vertebrate conditions and how those changed in those respects. So we begin therefore with an Agnathan, which is a lamprey. It's a blood-feeding parasite. So it's got quite a specialized derived state, but still reflects the early Agnathan jawless fish condition. So the emphasis here is really to recognize in the external anatomy, we can see that we have our external gill slits, because this has the respiratory system, but it's also going to be sucking blood, and so it's going to have a digestive system and needs to partition those. And so the major theme is that they have a velum for doing so here, which allows the partitioning of the esophagus and the respiratory tube. So if we focus in on the respiratory system, there would be the emphasis on the brachial baskets with the internal and external gill slits to enter that and to leave that. Then with respect to digestion, it's really the emphasis on how simple their digestive system is. It's really a linear tube without any differentiation. They have some cells in the wall that constitute a pancreas, but nothing more. The other major components is to think about other aspects of their integument. Really, they have the myomeres and the myoceptor, the notochord, allows that undulatory movement that we see in this species. So it's a body caudal fin BCF swimmer. And then on top, which you can see a few structures, we have here the nostril, and then we have adjacent to that the pineal gland, which is a light-sensitive organ. In that, it later becomes the pineal gland in the latter vertebrates for controlling circadian rhythms, etc. So that makes some sense. We also have a epiphyseal sac within, which can be identified in, I believe, this particular cross-section here. And the epiphyseal sac is actually can become your pituitary gland, and that one is derived from the Hatchek's pit in the cephalochordates. So we can see that charting forward. Okay, and then there's a good opportunity to see the respiratory structures here and make sure you know those. All right, we're going to jump over this, and we're going to jump to our Chondrichthys, which represent a gnathostome, a jawed fish, and an early branch, which is highly derived but still represents that state. So this is an active predator with a cartilaginous skeleton. And if we look at our respiratory and digestive system, these guys do not have the internal gill slits, but they do have the brachial basket and the external, or the hemibranch, the brachial pouch, with the external gill slits. And there's 5 to 7, depending on the species, and it does vary. So multiple gill slits. So with these guys as well, they have spiracles externally, but you can look at my external dogfish anatomy in a separate video, which I'll make, along with the detailed internal anatomy, because this specimen is rather aged, so it's better to use a new one. So the emphasis will be on the differentiation of the digestive tract as they now gain a higher degree of specialization to increase efficiency. What's amazing in this image, though, is that we can also see that this is a female, and it's not the emphasis of this lab, but you can see that this is a baby shark, little baby shark, because this has actually got one of the longest gestation periods of any vertebrate. It's an ovoviviparous, so eggs retained internally where they develop, and gives birth to live young. So it's quite an astounding thing to see there. So up to 24 months gestation. And that's just a quirk of this particular individual. Kind of a shame, really. Hopefully we would prefer not to capture old females for labs. Okay, so we'll move on round now. And we're going to think about this fresh dissection station. Look at the 2 videos on external and internal anatomy for the dogfish over here. All right, we then come to our Mudpuppy, and this represents really the tetrapod condition, so a more derived vertebrate, and we're again seeing that increasing specialization of the digestive tract, and so look for the various structures that contribute, and particularly the differentiation of the intestines into a small and large intestine with multiple subsections of the small intestine, and the increasing length and therefore surface area of that structure. We also examine the respiratory system and recognize that this is nearly a fully aquatic species. It's a mudpuppy, and therefore our mudpuppies as a nearly fully aquatic active hunter of invertebrates and small fish, they actually retain an external gill as a result of pedomorphosis. You can see it better here, but it's also pinned there as number 8, I believe. And so this external gill, also number B, coupled to the skin, C, and then the lung, A, allows them to respire in 3 different ways. These guys, because they're mostly aquatic, gain very little oxygen from their lung. So that's the story of them. So okay, this is the early vertebrate dissection, respiration and integument lab, and we're just going to have a look at the integument here at station 4. So this theme here is about the origin of scales, so that's from dermal armor, which then in our earliest fish forms the placoid scale seen in Chondrichthys, which is then going to be bone with an dentine overlay and then enamel, and these are called the placoid scales. So the placoid scales form denticles which reduce skin drag by breaking up turbulence, creating a fine boundary layer in sharks. Now those then modified to what's called a cosmoid scale, which is a flattened, more classic looking fish scale, which again is bone, dentine and enamel. Those are only seen in coelacanth and lungfish today. Most other fish have modified further with reduction, the loss of the dentine layer in the ganoid scale, which we see in early fish like gars and bowfin, and so that's again a lighter skin once more, lighter scale. And before we go to a super light scale in the cycloid and ctenoid scales of the derived fishes, the bony ray-finned fishes, particularly teleosts. And this one is actually just composed really of a very fine bone layer with a sort of cross network of connective tissue and very fine enamel layer. So we've seen a trend towards dermal armor to the super light scales, allowing much greater mobility, flexibility, and lighter as well, because these are going to be less dense, so that's going to aid in buoyancy. Now we go to the tetrapods, and the amphibian is missing here because they basically lose scales. And that's because they're using their skin for respiration on land, but it does mean that they then can't go further into land because they need to keep their skin wet. So we saw the re-evolution of scales in the amniotes coupled to their egg. So those are adaptations for dry land, but it's convergent evolution on scales because the early reptilian scale is not derived from mesoderm and bone, it's derived from epidermal keratin. So this epidermal derivatives make scales. They make the cornified epithelium of keratin in a human as well, and they also make hair and feathers. And in some structures they interact with the underlying dermis, which is mesodermally derived, to create bigger structures too. And so we can go and have a look at this in our station 5. So here you're just seeing it's the epidermis and the dermis with the basal lamina between the two as a membrane. We move this way to station 5, we see all of the. So here we just see the epidermal derivatives that are keratin-based from a cornified epithelium. So we're talking really the scales, hair, feathers, claws, baleen, these kinds of structures, some of which are also combined with mesodermal derivatives to make more complex systems, like, for example, a horn, which combines bone with overlayer of the keratinized horn layer. So we see other complex structures there. And these are all in the amniotes.